Okay, hi, welcome to another Soul Seminar Online. I have a couple announcements. First and foremost, I would like to thank Andreas for all his help being a co-organizer over this past year. Due to some scheduling conflicts, he's stepping down, but we have some exciting news that Luke Wheeler will be on our committee, our coach in helping us co-organize for the time being. And also we have announcement, our next guest host will be Matt Gibson. I think they're both on the call. So welcome, and we're excited to have you and help us organize. We do have a short list for inviting speakers for our second session. However, we will have a Google Forms that if you're interested in self-nominating or nominating other people, that you can do that. That will be on the website, and we'll be able to email that out a little bit later. Cool, so we'll go ahead and get started for this week. So first, I would like to introduce Amanda Broz. She earned her BS and MS in plant biochemistry at the University of Missouri-Columbia, followed by a PhD in horticulture at Colorado State University. Over her career, she has worked with diverse projects with the goal of linking molecular and chemical signatures in plants to physiology and evolutionary outcomes. She's become interested in Solanum and the evolution of plant mating systems during her postdoc work at the laboratory of Pat Bedinger. She is currently working as a research scientist at Colorado State University. Next, we also have Pat Bedinger in this talk. So Pat did her PhD in biochemistry at the University of California, San Francisco, studying the enzymes of DNA replication, and began studying plants as a postdoc in Stanford University. Pat now has her own lab at Colorado State University, where she switched from studying plant molecular biology to studying the evolution and mechanisms of reproductive isolation barriers between species of wild tomatoes. She, with the support of NSF and Fulbright Scholar Award, she became very late in her career to do field work in Peru and Ecuador, and she wished she was doing it her whole life. With that, you can go ahead and start.
Okay, thank you, Chelsea. We'll see if this works. Can you see anything yet?
Not yet.
Oh, okay. I need to go back and. Oh, sorry. Where is? I need to find. Oh, there it is. Share screen. Yeah, does that seem okay?
Perfect.
Okay, great. User pointer. Okay, great. Well, it's lovely to be here. And I'm the person that keeps asking about mating systems and sympatry with your species. So it's really fun that I get to tell you why I'm so interested in those things today. And obviously we're working on the wild tomato species of South America. But I wanted to start with thanking some very special people and places. And that includes anybody who works in wild tomatoes. Has a great debt of gratitude to Charles Rick, who not only made many, many excursions to South America to collect accessions of wild tomatoes, but he was also instrumental in starting the Tomato Genetics Resource Center that provides seeds of all of these accessions for free to anybody who's doing research on them. So all around the world, people use the Tomato Genetics Resource Center, which has been really a wonderful resource. Another group I want to thank is the National Science Foundation. And when I decided to work on this project, I started just calling people up and saying, would you guys like to work on species barriers in tomato? And I talked my my three science buddies into joining me. So Steve Stack, who's a wonderful cytogeneticist, Bruce McClure, who is the co-discoverer of S-RNases, and Roger Shedley, who is now the director of the Tomato Genetics Resource Center. And through the years, we've had various iterations of this group, and this particular one includes Leonie Moyle and Matt Hahn, as well as our groups. I also want to thank the Fulbright Foundation for letting me go live in Ecuador during my sabbatical, and Universidad Particular de Loja for hosting me, and Pablo Acosta was my host, and my field assistant, Tanya Rio Frio. And finally, I want to thank this series. I have just loved this seminar series, and ever since I found out about it, I haven't missed any of them. They're just really so wonderful, and I want to thank the organizers for this. Okay, so compared to the clades that I've been hearing about in this series, this is sort of a tiny clade, the tomato clade, which does include 1 cultivated species and 12 wild species. And I chose this group to study because of the variety of mating systems among these species. And I'm going to say SC and SI a lot. That will mean self-compatible or self-incompatible. And you can see that there are a number of species that are self-compatible. About half of the species are self-compatible. And the other half are self-incompatible, but have some self-compatible populations that are more recently evolved. And those are going to be really important for Amanda's part of this talk. This is, if you go to the TGRC, you can now see where your accession was collected. And these are all the wild tomato accessions that are at the TGRC that you can access for free. And one thing you can notice from this is that the ranges of the wild species overlap. There's none that are completely isolated geographically. And many can be found in Sympatry. And through the TGRC, you can identify at least 38 documented cases of sympatric sites. So at one point we decided this is really wonderful that we're able to access these accessions, and we actually ended up going to Peru and Ecuador and finding many of these as well that are still present. And we selected 12 sympatric sites. We were careful to combine all of the possible mating systems that we could, 2 SI, 2 SC, 1 SI, 1 SC, SI populations, and SI species, and all of the possible mating types that we could think of. We picked those sympatric sites to focus on. So just to sort of review in different possible interspecies barriers that are known, there are, of course, pre-zygotic pre-mating barriers that can prevent hybridization between species. And these, of course, include probably the most important is geographic and habitat isolation. But there can be, oh my gosh, so this is a little bit weird. It should say phenological isolation, where one species will bloom at a different time than another one. And of course, there's behavioral, which means pollinators in our case, and mechanical isolation mechanisms that can prevent mating. And then there are pre-zygotic post-mating mechanisms that will include something I'm going to talk about quite a bit, which would be gametic isolation, where the pollen tubes here shown in staining with the black in this case would never reach with the sperm, never reach the ovules with the eggs. And then, of course, there are post-zygotic barriers as well. And one that I'll talk about quite a bit is barriers that prevent seed formation in hybrid fruits. And there are cases, we didn't find any natural cases of this in our wild tomatoes, but there are in other species, there are post-zygotic factors where hybrid plants will exhibit hybrid sterility or hybrid necrosis. So in terms of sympatry, like I said, there are 38 known ones, but they're common. We discovered new sympatric sites when we went to Ecuador and when we went to Peru. Sometimes this was just this sort of traffic incident where our car couldn't get past another car and the scientists went off wandering like we tend to do. We found a site that had 3 species in sympatry. So sympatry is common with the wild tomatoes. And at every site we went, both or 3 species, they were all flowering simultaneously. So co-flowering is common at these sympatric sites. This particular one, oh, and I should say the way I'm defining sympatry is very, very strict. I have to be able to stand at one species. So I was standing over here by this Arcanum species and looking over here, and I could see the Solanum pimpinellifolium here. So I had to be able to see the other species as I was standing at one. So, but of course, pollinators can fly much farther than that. Okay, another thing I would like you to sort of notice about this picture is that it's clear that both species have been visited by pollinators from the anthercone bruising that we would see. And that leads me to the next possible barrier between species, and that is sharing of pollinators. And can pollinators transfer heterospecific pollen? I think probably, but we really, honestly, we don't know. This is sort of a big blank for us. We did, get some species of bees. These are all buzz pollinated. So we got some species of bees that we found on Solanum habrochaites, both in Peru and in Ecuador, and there's a number of them. And but we haven't done a careful study about whether heterospecific pollen gets transferred via pollinators. But next time, and I would advise you to do this as well if you're interested in having a pollination expert with you, is bring Maria Vallejo Marin with you. He's a wonderful, we should get him to give a talk because he's a wonderful researcher on pollinators, including especially buzz pollinators. And so he could help us fill in this blank. Now, one thing I want to point out is that this picture was taken in Ecuador and obviously being visited by a pollinator. And this particular plant is a self-compatible, part of a self-compatible population. So I have to keep reminding myself that self-compatibility does not automatically mean that there's no outcrossing going on, because we saw lots of pollinator activity among the self-compatible populations that we study. It's just something to keep in mind. And so going on to the next possible kind of barrier, and this is the kind we study a lot, are that there can be pollen pistil incompatibilities. And in many plant families, not just the Solanaceae, these kind of incompatibilities can follow the SI by SC rule, and that's also known as unilateral incompatibility. So what this means is, so I have an example here of Solanum pinellii, self-incompatible, trying to be crossed with a self-compatible Lycopersicon. And you can see that pollen tubes can grow in this kind of cross, but not in this kind of cross. And that's what's shown here is staining of pollen tubes. If the self-compatible species is the female in this kind of cross, pollen tubes get all the way down to the ovary. But in the reciprocal cross, if the female is self-incompatible, pollen tubes stop growing very close to the top of the style. And this is actually a faster stop than in self-incompatibility. So this is the one thing we have been studying a lot as a group. And one of the first things we did was just assess this kind of behavior in all of the tomato species. So graduate student Yusun Beck crossed all 13 species as male and female with each other. and to test whether the SI by SC rule was followed in the tomato clade. And all this shows right here, I'm not going to have you read this, but all of the species is male and all of the species is female. And what she found was that when the female was self-incompatible, pollen tubes were stopped of self-compatible species. So the red is stop, reject. And in contrast, when the same species were used, but the self-compatible species was the female, pollen tubes reached the ovary, and in some cases set seed, which I'll talk more about that later. These didn't set seed, but the pollen tubes got all the way to the ovary. And then with like mating systems, in general, they were compatible SI by SI or SC by SC, with a couple interesting exceptions that we're following up on. This is really a puzzle that we're exploring still, where it didn't fit this pattern. But so, of course, these were done in the greenhouse. These crosses were done in the greenhouse with accessions that we got from the TGRC. So we really wanted to see whether this was relevant for natural populations. And what we found, where in some of these crosses were actually done in Peru, what we found was that, yes, indeed, sympatric populations could exhibit this SI by SC rule if the female was self-compatible. So you can see that pollen tubes are stopping very, very close to, they germinate just fine and start growing the pollen tubes, but they don't get very far in the style before they're rejected. So we found that kind of cross occurring, well, could occur at 5 different sympatric sites. We found 2 other kind of interesting pollen pistil interspecific barriers. So in one case, there was a pair of self-incompatible species, and what happened is in the heterospecific cross in one direction, the pollen tubes grew much slower. So they took over 3 days to reach the ovary, from the stigma to the ovary, whereas the conspecific pollen tubes would reach the ovary in less than 24 hours. So we saw that in one case, but it seemed to be, it didn't depend on whether it was the sympatric accessions. It was just those 2 species combinations that would happen. And then in another 2 cases, we actually found that pollen tubes would go all the way down to the ovary, but then they would just sit there and not target ovules. So the lack of ovule targeting. And in these cases, you wouldn't get a fruit. So you have to get fertilization to get a fruit, which leads me to the next type of interspecific reproductive barrier that we found. And that was now post-zygotic. So we got fruits, but the fruits were a bit smaller in the cross than normal fruits for those species. And when you looked at the seeds, they were much smaller than normal seeds. So these are the same magnification. So we took a much closer look at those seeds. And this work was done by wonderful microscopist Suzanne Royer. And here I'm just showing normal tomato seeds that are almost entirely filled with the embryo, and they have a little bit of endosperm left. But this is, I'm showing an earlier stage in seed development here, just because I want you to see what a younger embryo looks like and what the endosperm looks like here in normal seed development. So this is an embryo that it's globular, just sort of round, and you can't tell where the cotyledons are eventually going to form. So it's just sort of round, and it's just called a globular embryo. And the Solanaceae have sort of an unusual endosperm development that's called cellular, and that's compared to most species. you don't get cell walls forming in the endosperm, developing endosperm until later in development. But here in our species, our solanaceous species, you have cellular endosperm, and it's really rapidly dividing at this time to nourish the growing embryo. Okay, so when we looked at those small seeds, and this is just normal to compare with, what we saw is they were blocked at the globular embryo stage. So you can't really see any parts that are going to become the cotyledons here. It's sort of a symmetrically round embryo here. But what we saw was that the endosperm didn't develop. So you can actually see collapsed endosperm cells here and here. But there were two different phenotypes, so it was really interesting. One is that we just saw breakdown of the endosperm, and all the other cell layers looked pretty normal in the ovule. But in some, and I think this is really interesting, in some cases, it depended on the direction of the cross, you would get a tissue that's called the endothelium that's usually one cell layer thick, dividing like crazy to form this overgrown tissue here around the embryo. And this had been reported like decades ago in Nicotiana crosses, and they actually called it a tumor in these hybrid seeds. So it appears to be the endothelium that is out of control here. And finally, we actually saw in our 21 crosses that we did, we found the formation of normal seeds and normal looking hybrid plants. Here's mom, here's dad, and here's a hybrid on all of these. And they had, somewhat, they looked different from their parents. Sometimes you could say, well, it really looks intermediate, or this looks just pretty different, or but the seeds were normal, the plants were normal. We really didn't follow up very much and go to the next generation, but they certainly produced pollen. So this was sort of surprised because hybrid plants have never been found in sympatric sites for wild tomatoes. So these particular three places, we should look harder, I think. And it's nice because we actually had markers where we could follow and prove hybridity. These are S-RNase alleles associated with different species. And so we could really tell that, yes, those are indeed hybrid plants. So to summarize this part, interspecific barriers in sympatry, we found, 8 cases of pollen pistil barriers that would prevent hybrid plants from forming, in 5 cases of the classic SI by SC rule. And then we found 11 cases, so about half and half pre-zygotic and half post-zygotic cases. And we had these 2 sort of evenly distributed phenotypes, one of which had this endothelial overgrowth phenotype, which I think is really fascinating. But out of all those, we found 3 cases where apparently interspecific barriers were lacking, and we could actually find viable seeds and viable F1 plants that we could prove were hybrid. So that was a really interesting study just to see what could happen at a sympatric site. And I'm going to now turn the presentation over to Amanda, who's going to talk about it. So a different thing we're really becoming fascinated with, and that is how mating system transitions from ancestral SI to self-compatibility, how that impacts population structure, and also how it can change interspecies interactions. So she's now I'll stop sharing my screen, and Amanda will take over with that part of the story. And we did these studies in our favorite wild tomato species, Solanum habrochaites.
All right, does my screen look okay?
Perfect.
I'm gonna hide you guys. Hi, everyone. Thanks for coming. My name is Amanda Broz. And like Pat said, I'm gonna talk a little more about mating system transitions in a particular species, Solanum habrochaites. And I've kind of framed this talk as what are the causes of mating transitions, and then what are the consequences. So like Pat mentioned, this is our clade, and it's extremely useful for us to help look at questions about mating system because we have all of these transitions from self-incompatibility to self-compatibility. They're very common. And we have these really interesting species in which we have many populations that are self-incompatible, but we have a few populations that are also showing self-compatibility. And so these are really interesting to kind of look at these questions. So we used Solanum habrochaites as kind of a test study case to answer the question, what are the causes and consequences of mating system transitions? So the first thing we did was assess the distribution of mating types. And down here, I'm showing a map from the TGRC, kind of like Pat showed, but this one only shows Solanum habrochaites. And as you can see, the range is from central Ecuador to south central Peru. So we chose about 40 accessions throughout this range to look at mating systems out of about 100 that are housed at TGRC. There are also additional accessions housed at USDA. And then we wanted to know what are the genetic and ecogeographic factors that lead to self-compatibility in this system? And then how does the transition to self-compatibility influence things like floral morphology, population genetic structure, and pollen-pistil interactions within and between species? So our first step in the process, obviously, was to look to characterize mating system. And again, I'm just showing the map kind of zoomed in here, looking at the range of our species of interest. And here I've made a simplified map and I've overlaid it with the mating types that we found. And so in black circles, we see self-compatible populations. In white circles, we have self-incompatible populations. And in some populations, we found a mixed mating system. So in some cases, individuals were both self-incompatible and self-compatible. And in other cases, we found self-incompatible individuals that would occasionally throw off a self-compatible fruit. So it's quite variable. Mating system is really labile, I think, in many species and in this system as well, obviously. So what you can see from this distribution is that we found SI, self-incompatible, and mixed mating populations here in the central part of the range. And this really agrees with the proposed center of origin for the species, which is here in the Cajamarca area around central Peru or northern Peru, I suppose. In addition, we found that SC populations occur at both the northern species range margin and the southern species range margin. And this is a pattern that people may be familiar with. So this pattern of self-compatibility being at species range margins has been observed in many species, and I think was really kind of put forward in the literature back in 1955 by Baker. And so Baker described a situation in which selection for self-compatibility would happen when mates are limiting. And we can think about this kind of using a little cartoon here. So let's say we've got a population kind of here up here at the, where the species is going to start migrating into a new area, and it's filled with self-incompatible and self-compatible individuals. There's some kind of dispersal event, be that to just, a little bit north, maybe a new island in some cases, things like that. And if we have a self-compatible individual, in an extreme case, say just one individual, that individual is basically going to go extinct. It doesn't have anyone to mate with, and so that's the end of that dispersal event. However, if we have a self-compatible individual, it's quite possible that through self-compatibility, that particular individual can reproduce and establish a new population. And so this trend has been seen quite a lot in the literature, and it's obviously Baker's law has been called a law now, although there are a lot of subtleties to this law, definitely. So next we wanted to look at what are the causes of mating system transitions to self-compatibility. And what Pat's lab has been really interested in addition to Bruce McClure and Roger Chatelet, is what is the mechanistic basis? So in the Solanaceae, we have a system where we have S-RNase based SI. And so let's look a little closer at that. So we have a self-incompatibility locus, or the S locus. And this is where these S genes are harbored. So in this locus, we've got, it's kind of shown here in a diagram in a cartoon. We have pistil expressed S locus RNase, or S-RNase, as hence S-RNase-based Si. And there is just a single S-RNase at any given S locus. And then we have this suite of pollen-expressed S locus F-box proteins, or SLFs, shown here. And so really when we think about this, we kind of regard the S type as a haplotype. So it's really an S haplotype that encodes a single S-RNase and the suite of SLF proteins. And generally there's around 20. So in this case, we can say this is an S1 S-RNase, and we're going to call this the S1 haplotype. So in any individual plant, for example, we're going to have two haplotypes, right? So we'd have an S1 and S2 haplotype, for example, in a given individual. And within a population, we're going to have many S haplotypes so that these individuals can mate between the population. So something interesting about S-RNase-based S-RNase-based self-incompatibility is that it works through collaborative non-self-recognition. And so this is a little different than the other systems of self-incompatibility that have been characterized. So for example, in Brassica and in poppy, we have at an S locus, one pistil side factor and one pollen side factor. And they basically kind of interact through, your classic lock and key mechanism. So they come together and you can recognize a self interaction. So self recognizes self, and that causes the rejection of self pollen tubes. In the case of S-RNase-based Si, we're seeing something different. So we see non-self recognition. In this regard, this suite of S locus F-box proteins is going to recognize and detoxify every other S-RNase except the one that's found at their same S locus. So this cartoon shows kind of a simplified version of what that means. So basically you can see that this S1 haplotype is encoding this yellow S1 S-RNase, and it is missing this, what is designated here as an anti-S1 SLF. And so in this sense, pollen from this haplotype cannot detoxify the S-RNase from S1, but it is able to go through styles that are expressing, for example, S2, because it has this anti-S2, or S3, anti-S3, and so on and so forth. And I say this is really a simplified model because, as you can imagine, if there's only 20 SLFs, then one S haplotype would only be able to mate with, a maximum of 20 different females potentially. But really what is happening, we think, is that multiple SLFs are getting together, and that interaction recognizes particular S-RNase proteins and detoxifies them, allowing pollen tubes to grow and have a compatible reaction. So in Solanum and most plant species, in fact, we have a gametophytic system where we have a pollen here. So this is just expressing basically one haplotype or one suite of SLFs, for example. So this S1 pollen is just expressing SLFs from the S1 haplotype. And this grows down through the diploid tissues of the style, right? So we have an S1, S2 plant here. On the female side, it's expressing the S1 and S2 S-RNase, but the pollen is expressing just a single haplotype. So in this case, S1 and S2 pollen are rejected. They're not capable of detoxifying their own self S1 and S2 S-RNases. But let's say, this individual that we can't see on this picture, S3 pollen comes over from a pollinator. This is able to successfully traverse the style. And we can think about this kind of in an architecture, wherein we have pistil side barriers. So for example, the S-RNase, and that has to be matched by specific pollen side resistance factors. And so I already mentioned, so we have our specificity components on the pistil side, that's S-RNase, but we know that there's other proteins that are critical in the self-incompatibility reaction. And so we call those modifier proteins. And one of the best characterized is HT protein on the pistil side. We've got a similar situation happening in pollen where we have specificity, which is determined by these SLF suites, but then we have modifiers as well. So for example, colon-1 is part of the E3 ubiquitin ligase complex. And this is important in degrading that S-RNase so it doesn't act as a cytotoxin to kill pollen tubes. So we can think about, thinking about this architecture of self-incompatibility. What are the pathways to get to self-compatibility? And so basically we can think of, mutations happening in either pistil side barriers or pollen side resistance mechanisms. And so there's been some mutations that have been characterized I'd say the most common that we see is a loss of function mutation in S-RNase. So you can imagine, let's say we have this same plant here, something gets messed up in the S1 S-RNase. In this case, pollen that is S1, expressing the S1 haplotype, is not exposed anymore to the S1, to its self S1 S-RNase. And so this pollen tube is going to be able to go down and reach the ovary and have a successful pollination. The alternative way in which we can get self-compatibility is through a gain of function in an SLF. And so what do I mean by gain of function? Basically, this would be a mutation wherein this S1 haplotype gains some kind of, or has a mutation in an SLF, and that allows it to now recognize its own self S-RNase and detoxify it. So in this case, it would gain the capability of recognizing S1. This pollen tube would recognize and detoxify the S1 S-RNase, and again, we would have successful pollination. So this is not as well, or this hasn't been found as frequently. It has been characterized in Solanum neorickii in one case, and also in Petunia. But typically we're seeing a lot of loss of function in female factors. And so that's what I'm going to focus on for this next part of the talk. But pollen side resistance is really going to come back. So we can think about this architecture in kind of a stepwise breakdown of pistil side barriers and pollen side resistance. And the shuttle lab has done a really amazing job characterizing some of these pollen resistance factors, and that'll come back into play later in the talk. So using this, or looking at pistil side barriers, the first thing we did was to do immunoblots just to look for S-RNase protein in our different populations. And so I'm just splitting this into two parts, so kind of the north and the south, starting with the south, obviously, so we can kind of look at what's going on. So in this case, I just basically took styles from flowers from all of these different populations, smashed them up, extracted proteins, ran a gel, and then did an immunoblot here with S-RNase. And so as you can see, in this case, everybody is expressing S-RNase. It doesn't matter if you're self-incompatible or self-compatible, everyone has S-RNase protein in styles. However, we also had some additional information. So a population down here, the furthest south accession of Solanum habrochaites, an allele had been characterized that encoded a loss of function S-RNase in a previous paper, so from 2010. So we extracted genomic DNA from all of these plants and looked for this allele. And what we found was pretty striking. So every population that was self-compatible contained this low activity S-RNase allele. And in this case, where we had a mixed mating population, we actually saw, I'm showing just a representative of a self-compatible individual here. So the self-compatible individuals in this population also contained this low activity S-RNase allele, whereas the SIs did not. Or self-incompatible individuals in that population did not. And so this is basically, you know, the distribution that we see of this particular loss of function S-RNase allele. We also screened all of these populations for HT protein. And as you can see, we see HT protein is present in all except for one population. And so this indicates that it's not a loss of HT that's leading to self-compatibility. It appears to be linked to this low activity S-RNase allele. So here in the south, we saw self-compatibility was correlated with the low activity S-RNase protein, but not a loss of S-RNase or HT expression. And we saw a single population had lost HT protein. So when we look at the northern populations, we see a really different story. So this is a blot basically from north to south. These are the self-incompatible plants from Ecuador, and these are the self-incompatible plants from Peru. So it's not exactly ordered north to south. But when we do a blot here, and we look for S-RNase protein, we see that self-compatibility is associated strictly with a loss of S-RNase protein. Interestingly, in these mixed mating populations where we see some self-compatibility, we don't see a loss of S-RNase protein. So again, we were lucky in that previously an allele had been characterized in this population here that encoded a loss of function S-RNase allele. And in this case, there was no transcript expression and no protein expression in this particular population. So we screened everybody for this particular allele, and we found that this known loss of function allele was present in a subset of these northern populations. And so I've shown them here on this map. So at the very northern range margin, we see self-compatibility is associated with a loss of this particular allele. Again, we screened for HT protein, and we found one population, interestingly again, that did not have HT protein. But we can see here that self-compatibility in the north is associated with a loss of S-RNase expression, and again, not a loss of HT. In addition, mixed mating populations and individuals that we tested all expressed S-RNase. So something else is going on here that's leading to this lability and self-compatibility. And a single self-compatible population had lost HT protein, this one here in the north. In addition, the specific loss of function S-RNase allele was found in a subset of populations. So based on that data, we could say, something else is going on in the north. We've identified one loss of function allele that's associated with self-compatibility there, but it seems that there must be others, right? What else is leading to a loss of S-RNase in those populations? So we went through the whole range and characterized new loss of function alleles using both degenerate primers and then transcriptomics. And so what we found is, as I mentioned before, all of these have the allele that encodes a low activity protein. And again, we found this segregating in our mixed mating population in the south. In the north, we had this allele that was not expressed, but then when we went looking for other S-RNase alleles, we identified some that were found in, for instance, this corridor around LOHA. This was just a very low expression S-RNase, so we could detect very low levels with transcriptomics, but it's obviously not detectable at the protein level using our system. We found one that just has a nonsense mutation, so an early stop codon. In another case, the S-RNase appears to lack a glycosylation site, which is probably essential for activity. And then we have other uncharacterized ones that we're still looking at. So cases where the plants are SC, but they don't have any of these other alleles that we've characterized yet, as of yet. And so we can see that Solanum habrochaites has undergone at least 6 transitions to self-compatibility. And we see this interesting pattern where we have 1 transition at the south and at least 5 in the north. So something else we wanted to explore as far as a cause of mating system transitions was the unique ecogeography of this area. And so I know a lot of the Solanaceous species occur in this area, and so probably many of you are really familiar with this. Here I'm showing a topographical map of this same region. And as you can see, we have the Andes here highlighted in gray and white. And in this particular region, we see basically a breakdown in the Andes. And this is a biodiversity hotspot for many different types of plants and animals, and it's known as the Amotape-Huancabamba zone. And so it's really interesting that we see, this is actually very close to the origin of Solanum habrochaites here. And so as it migrated through this unique habitat zone, it looks like self-compatibility arose multiple times. And we can think about this, using this cartoon for Baker's law. So let's say, we've got these populations here, and maybe mates are limiting just due to, the plants being self-incompatible, or maybe there are changes in pollinator or habitats in this zone. This area is associated with many diverse microhabitats. So when we have a dispersal event where the species is traversing through the AHZ, it appears that we're seeing recurrent selection for self-compatibility in the north. And it's likely due to these ecogeographic factors. So in summary, for causes of mating system transitions, we see in the southern populations, self-compatibility is associated with a specific S-RNase allele that encodes a low activity protein. And that haplotype has successfully spread in the south and is fixed in nearly 1/4 of the species range, so very successful. In the north, we're seeing something different. SC is associated with a loss of S-RNase protein expression, and at least 5 different loss of function alleles have led to self-compatibility. And this may be due to the diverse and isolated habitats in the north, which have probably led to recurrent selection for self-compatibility. So let's look at some consequences of these mating system transitions. So in many cases where we see plants going from self-incompatible to self-compatible, it's often associated with this Sulfine Syndrome, as it's called. And so we see plants, the flowers they don't need pollinators anymore, and they take on a lot of selfing, so they become smaller. The stigmas are not exerted, they're recessed inside, so they can be immediately pollinated once pollen is available in the plant. So we looked at this a little bit in the northern populations, and I'm kind of showing over here just some representative examples. And so we did see there's a huge variation really in some of these SC flowers. But in general, we saw that self-compatibility was associated with smaller flowers, and this is showing just 2 different times. So when the flower opens and then the oldest flower on the plant, So self-compatibles are smaller than self-incompatibles in both cases. So it looks like there's some, characteristic changes of the Sulfine syndrome happening, but we didn't see any reductions in stigma exertion or other characters. And as Pat mentioned, just looking at these plants and these populations, we saw pollinators everywhere and bruising on anther cones. So it seems like there's still a lot of interaction and probably crossing going on, at least within a population. Something else we were really interested in looking at was, what is the population structure? So we've looked at loss of function S-RNase alleles, but one of the graduate students in Pat's lab was really interested in looking at genome-wide neutral markers. And so to do this, he collaborated with Gaurav Moki and Jacob Landis over at Cornell as part of a larger RAD-seq experiment. And what I'm showing over here is just the structure plot, basically going from north down to the south for different groups. And as you can see, it's a little, so this yellow bar here represents the Ecuadorian populations, and this red bar represents the Peruvian populations. And over here, I'm just showing a map of all of these neutral markers that are showing how populations basically clump out genetically and where they're located on the map. So for example, this most northern one is shown here, and the pie is showing kind of the distribution of genotypes that are associated with that. So we can see down here, we have these self-compatible populations down in the south. They're all associated with this purple genotype. We see orange and here in the north associated with this particular genotype and self-compatibility. We see multiple self-compatible types here. And this was really exciting because in this case, we're looking just at genome-wide neutral markers. And if we put this map next to the map we generated for our loss of function S-RNase alleles, we see that we have a lot of parallels. So it looks like a single transition to a loss of function S-RNase allele is really influencing population genetic structure in this case. So I've tried to kind of color code these in the same way, where this orange loss of function S-RNase allele correlates well with these orange pies and this genetic structure. And similarly down here in the south, the purple and the green. In addition, something really cool that Gaurav was working on was looking at acyl sugar diversity in this system. So secondary metabolites in trichomes and leaves. And we found a lot of overlap between fixation for different acyl sugar alleles, so that change is acyl sugar diversity, and our mating system types. So for example, we could see as the species migrated north through the AHZ here, it looks like they're accumulating this one change in that causes differences in acyl sugars. And up here we see fixation that's associated with this SC or genetic group. So this was really cool. It seems like SC is really associated with population differentiation in this structure, in this species. And we can see this, using our neutral genome-wide markers, looking at S-RNases and looking at other traits. So those studies suggested that SC groups are differentiated, and we do a lot with pollen pistil interactions. So one thing we wanted to ask is, are there emergent reproductive barriers between populations? So to do this, we really focused mostly on the north. And here I'm just showing you the map I showed before. So we've got mating system, designated as previously. And so what we did for our first study was to take a pistil parent, so a self-incompatible parent, and we took pollen from all of these different populations and put it onto this pistil parent. And we also did a subset in Peru. So this is the farthest south Peruvian accession down here that I will show that's self-compatible. And so, we can have two kind of hypotheses about what's going to happen, right? So one is, well, they're all the same species, so they should all be able to freely mate with each other. The other is, we know about the SISC rule, like Pat talked about, so possibly mating system transitions to self-compatibility are going to cause a loss of pollen resistance factors that's associated with an inability to traverse styles of SI pistils or populations. And so I'm going to lay over on this graph, basically the interpopulation response. So we either have pollen that's accepted by this population, pollen that's rejected, a broken heart by the SI population, or a population that's segregating. So individuals had different responses. So our expectation, right, is that if we put SI onto SI, we should definitely see pollen tubes being accepted by this pistil parent. And that's exactly what we see. In addition, we see mixed mating, all the mixed mating populations, their pollen is accepted by this self-incompatible parent. When we put self-compatible pollen on, we see a subset of these are accepted by this pistil parent, but others show segregating phenotypes or a total rejection of pollen tubes. So pollen tubes of all of these individuals from all of these populations were rejected by this pistil parent. Interestingly, we also see the same thing when we apply pollen from this furthest most south population to this pistil parent. And so this has been documented as well in some work by the Chatelet lab, as I mentioned, and some of the responsible factors have been identified, particularly in the southern population, so the loss of pistil side resistance factors. But as we can see, this is really interesting because an interpopulation barrier has been formed. So we have SI populations that reject pollen from a subset of our self-compatible populations here. So something else we wanted to test, just to kind of expand the study, was take pollen from this parent, so one of the ones that was rejected, and apply pollen to pistils of all of these different populations. And so again, here's our key. So either these pistils will either accept this interpopulation self-compatible pollen or reject it. And we already knew that, this guy down here rejects pollen of that one. And similarly, all of the self-incompatible populations, as well as the mixed mating populations, reject pollen from this parent. When we applied pollen from this parent to the self-compatible populations in the north, we saw that all of them accept interpopulation pollen here, suggesting that maybe a lack of S-RNase is associated with the acceptance of this pollen. And it doesn't need the pistil side barrier or the pollen side resistance at all to traverse these pistils. And interestingly, when we put pollen on the southern population, we saw something that had been noted in the past back in 1991, that pollen of this particular parent is rejected by self-compatible population in the south. So again, we see this interpopulation barrier is formed. So self-incompatible, mixed mating, and southern self-compatible populations actually reject pollen from this northern population and actually extends to this subset, all of the same ones that were fixed for that allele. They're that specific loss of function S-RNase allele, they also are rejected by all of these different groups. So the transition to self-compatibility impacts interactions within the species, but what about interspecific reproductive barriers? So to test this, we used Solanum habrochaites, all those populations as female, and we put on pollen of 2 different self-compatible species. So either the cultivated tomato, Solanum Lycopersicon, or a close wild relative, Solanum neorickii, that's also self-compatible. And when we did that, we either saw one of these 3 responses. So rejects interspecific self-compatible pollen, accepts S neorickii, or accepts S Lycopersicon. And so again, based on the SI by SC rule, we're expecting all of the SI populations are going to reject these SC interspecific pollen tubes. And that's exactly what we saw. Similarly, mixed mating populations reject. We saw many of the central corridor populations, they all rejected interspecific self-compatible pollen, but we have this subset that accepts pollen tubes of S. Solanum neorickii, and in addition, this one that accepts pollen of Solanum Lycopersicon, the cultivated tomato, which is kind of unheard of. That's like that means it basically has no pistil barriers, it seems like. And as you might remember from that plot, I showed one population in the north that was lacking HT protein. It turns out it's this population here. So this is lacking S-RNase and HT, and its interspecific barriers are obliterated, at least at this pollen pistil level through the style. If we apply pollen down here, we see that this population is still capable of rejecting all interspecific pollen. So here we see something interesting. A subset of self-compatible populations accept pollen from other species. We're seeing these species barriers are breaking down in this northern subset. So consequences of mating transitions. Let's just do a quick summary because we're running out of time anyway. So we have some southern self-compatible populations have smaller flowers. We saw that self-compatible transitions are associated with genetic differentiation, and between population gene flow can be further reduced by these emergent pollen pistil interpopulation barriers. So we saw central SI populations are rejecting pollen from subsets of both northern and southern self-compatible populations. And in some cases, we see this instance where species barriers are breaking down and self-compatible populations are able to accept pollen tubes from Solanum neorickii and in one case, Solanum Lycopersicon. And so I wanted to just kind of bring this back around to the architecture of self-incompatibility and think about the stepwise loss of genetic factors that are important for mating system transitions and likely for speciation. So we've got Si, our ancestral state, self-incompatible. Then we transition to mixed mating, so a partial loss of Si, and we're not sure, this is not associated with a loss of S-RNase, so it seems like it's a lot associated with a loss of some unknown pistil side factor. Then we have a transition to self-compatibility. Here we have a total loss of self-incompatibility, and this is associated with loss of function in a pistil side barrier, and that's S-RNase in all of the cases that we've looked at. In one case, S-RNase is still expressed, right? But it's a low activity protein, and so we would still regard that as a loss of function allele. And these specific transitions to SC are also associated with genetic differentiation. We have another additional case so that those far northern populations have the formation of an interpopulation barrier. And this is associated with the loss of a pollen side factor. And so the Châtelet lab has found that SLF23 and colon-1 are important for some of these pollen side resistance factors. There's some of the pollen side resistance factors that have been lost, but there are others that are still uncharacterized. Then we have this third case where we're seeing loss of interspecific barriers. And so this points to loss of pistil side factors. So in the one case that I pointed out, we have loss of HT protein. There are also loss of other unknown proteins. So Solanum neoricii was able to traverse styles in that northern subset, but they still all have HT except for the one population. So other unknown proteins are potentially involved in this response. So in summary, at least 6 transitions to self-compatibility have occurred in Solanum habrochaites, and they're associated with distinct loss of function S-RNase alleles. We see that ecogeographic factors facilitate recurrent selection for self-compatibility in the northern part of the species range, and that transitions to SC are associated with changes, especially in genetic differentiation and floral morphology. And, oh, there we go, sorry. Gene flow between populations may be further restricted by these emergent pollen pistil reproductive barriers. And interestingly, we see gene flow between species could possibly increase due to the breakdown of interspecific pollen pistil barriers. But, like Pat mentioned with the sympatric site, there's a lot of other stuff that's going on after the pollen tubes get into the ovary. I didn't look at seed set in any of these instances. So that could be another level that is where there are still barriers that occur. And so with that, I'd like to say thank you to Pat and all of my co-authors on this paper, in particular, Bruce McClure and Alejandro Tovar Mendez from University of Missouri. They did much of the Western blotting work. Chris Miller, Gaurav, and Jacob did most of the population genetics work, and then Roger Châtelet at the TGRC. And briefly at the end here, I'd like to give a quick plug to a colloquium that Pat has organized for Botany 2021. So whether you're going or not, you should come to our speciation mechanisms in plants, some colloquium. We have all of these different speakers and really kind of a diverse array of systems that we're going to be looking at. So I think it's going to be really exciting. And so with that, I guess I will end my show and
awesome. Thank you so much, Amanda.
Stop sharing my screen, and sorry for running a little over.
You're fine. So now we'll open up for questions. So if you have questions, you can either jot them down, make a note that you would like to ask a question. I think it's really great work. It's a lot of work and effort, and so cool that everything that you're able to investigate in this system. Really awesome.
Yeah, it's really a lot of work has obviously gone in on the backside. So the system's really well one of the best characterized, I guess, for self-incompatibility. And there are these, interesting overlaps between self-incompatibility and unilateral incompatibility.
Do you find that there's a big difference between like the number of S alleles? I don't know if you specifically looked at this or thought about this between like the northern and southern populations, and could that be like a big part of why you're seeing this? Is just because there's so many, maybe the F boxes have mutated quite a bit, or there's
so the S Locus so SLFs are actually rather hard to characterize. So there's a lot of F box proteins throughout the genome, obviously, and so that's one of the challenges. So even just using, one way is to look through pollen-based transcriptomics, for example, and get ideas of what S Locus F boxes are there. But really, the ideal situation is kind of mapping the S Locus and seeing like what S-RNase is there and what is this SLF suite that it's associated with. And similarly, I've done a little bit of work in the South. So in that population that was segregating for SI and SC, I characterized other alleles that are the S-RNase alleles that are functional and self-incompatibility, but we haven't, like how many S alleles are actually within a population is something that hasn't really been characterized well in Solanum habrochaites.
Yeah, interesting. Do you think there's been like bottlenecking in some of these? Okay, and then
so that's definitely a possibility. A lot of it, so in some ways the TGRC is an awesome and excellent resource, but in some ways we're limited by what the original collections were, right? So for example, the one I'm talking about in the South that's segregating, the original collection was only from 2 plants. And so you can think, it was a very large population and there could be multiple sires happening, and even only collecting 1 fruit, you might get this huge representation, but you might not. And so, that's a little hard to say. And, sadly, we can't really transport material back and forth anymore from South America. Like we, were able to do some field work down there, but we made some collections that are in South America, but we can't work on them here.
Yeah, understandable.
Currently.
Stacy has a question if you want to.
Yeah, that was all such amazing stuff. I wanted to say to Pat too, I was really impressed with, the crosses in the field, because I do always wonder how much our greenhouse crosses sort of replicate what happens in nature and how applicable it is. So that's awesome. I was also interested to see the morphological changes you'd shown. And, because of working with Chelsea, I've been interested in how, we often have talked about Selfing syndrome sort of at the macroevolutionary level, but then we see it happening within a species. And so I always wonder, how do those suites of traits come along so fast, and whether you think there's sort of standing variation for flower size and then it just erodes that way. I think it's probably not anything genetically linked, but it could be. So anyway, I'm just wondering what you think about sort of the speed and repeatability of selfing traits at the within species level.
Should I answer that one?
Sure, go ahead.
So Sulfine syndrome, it's thought that if you don't need to outcross, you don't need to attract pollinators. There's a selection for smaller and duller floral parts. But the other thing, I noticed while I was actually there is that these particular populations with the smaller flower size were in a completely different environment. They were typically, Solanum habrochaites is out on a mountainside and it's almost all by itself, but in those areas it was, deep embedded in lots of other plant types and a lot more herbivore pressure. So I think there's a lot going on there other than just the attract the pollinator story.
It's hard to say too. So I think something really interesting that we found, so I always, kept highlighting that little subset of northern populations, right? And that's the one where we see, this particular loss of function S-RNase allele. We see this particular breakdown of reproductive barriers. We see this particular fixation in Garov's work of this acyl sugar protein mutation. And we also see the small flower size. It's just within that one group. So, in this case, I think there's probably standard genetic variation. And maybe that just indicates that it's this happened once and then that one just spread. And possibly that's due to this, enhanced capability for selfing.
Thank you. I think Mario has a question. Do you want me to read it, Mario? I'll give you a second if you want to. There we go. Okay, cool.
I can ask it. Thank you very much to both of you for this really amazing talk. I really, really enjoyed it. I'm always I'm more and more fascinated by these wild tomatoes. And I was wondering if either of you could tell us a bit more about other ecological pressures besides this lack of mates or potential lack of pollinators that may be driving this recurrent evolution to selfing and self-compatibility.
Do you want to comment, Amanda?
I don't know. I don't think we have any good data to necessarily say anything. Obviously, just with those changes in habitat, there's a lot of other different pressures. So different herbivores are probably there. We saw some interesting damage on a lot of the plants in Ecuador, for example. A lot of the anther cones were eaten down, which is I don't think something that had been noticed in Peru. Is that right, Pat?
Yeah, they've just been clipped off. So yeah.
So even herbivore pressure onto the flowers, and other than that, just the habitats are quite different and diverse. So those ones in the north are actually like up on the coast, and it's much more humid and hot there. So I don't know. So those could definitely lead to different selection pressures, but we haven't really characterized anything. We left some like eye button things out to kind of look for differences in, you know, environmental factors, but many of them did not make it through the year.
Yeah.
There are a lot of landslides.
Something shiny that somebody picked up. You know?
And yeah, also probably people walking around grabbing things.
But altitude, if I had to say one thing, it would be altitude. Which with the accompanying temperature differences, yeah, major ecological change.
And sort of related to this is, have you, are there any difference in the persistence of these SI and SC populations within the species where, you know, maybe you recapitulate what the short-lived life of self-compatible lineages? Do you find that these transitions end up with higher probabilities of extinction or something like that?
That's certainly the conventional wisdom. Yeah, I'm not sure how much I buy into that, but we didn't talk about it. But one thing we do see is hybridization of the self-compatible populations. There's regions of Ecuador where they're hybridizing with each other. We can actually see that. And so, but we don't know if that's a possibility for rescue or not. I mean, certainly.
Yeah, I mean, we're looking over a short time scale too, right? Like Charlie Rick did an excellent job like documenting all of these populations and, you know, where they are located. And so Pat and myself, we were able to go back to those places and find both Pat in Peru found self-incompatible populations. And then in Ecuador, it's mostly the self-compatible populations that we were finding and in the same place, and then additional ones. So we found more. And when we couldn't go back, so we went back 2 years, and occasionally we wouldn't be able to find our population that we had before, but it was similarly because there was a whole landslide that like knocked off the whole hillside, for example. But we didn't really see any of them going extinct. In fact, we like found more than we expected, I would say. Luckily, they like disturbed areas, so it's good to drive along the side of the road and spot their yellow flowers.
Thank you very much.
I think Sandy has a question or comment.
So I was just going to say, those northern populations that are on the coast, it's not that it's hotter, it's that it's very cold in the winter. The fog comes in. That's a particular kind of habitat that's from that part of Peru all the way south to Chile, which is called lomas. And there's particular things that grow there. There's also subpopulations of arcanum, which grow in those lomas, and they're always a little bit different. But what I was going to ask is, did you find and try crosses between the populations of habrochaites, which are glabrous, and the ones that have hairs?
Oh, certainly certainly we have done that, but we typically looked at pollen tube growth rather than the next generation.
No, I'm just wondering, because if you're talking about acyl sugars and all that, like Stacy was talking about all that stuff going along, that's really different in those glabrous ones than it is in the hairy ones. It's completely different.
Absolutely. And so I think generally we see, except for those situations I pointed out, so the very most far south are like very hairy, and they're rejecting pollen tubes of that very small subset of northern populations, but we don't see them.
There's just a few in the north that do that. And the other thing I was going to say is that we've found, working with Galapagos tomatoes, that actually the things from the TGRC, and it's great that they're there, but they change in seed banks.
Sure.
Things change.
Yeah.
So the real test of all of it is going to be doing the crosses in the field.
No, I mean, that's why we made our collections in Ecuador and Peru. And it's really, I mean, I understand why the laws have been made, but it's really disappointing from a research standpoint that we can't.
I know, and it's hard to get stuff out as well. It's hard.
Yeah,
well, now it's hard to get stuff in as well. Yeah. Tomatoes we're no longer allowed to import them as well.
No, I know. Iris Peralta and I just gave talks at the Ministry of the Environment about tomatoes, wild Peruvian tomatoes, and biosecurity.
Yeah, it's sad.
Well, I mean, you know, it is a thing.
Good excuse for more field work. It is really
Yeah, it's a good
challenging
excuse for
though.
more field
So.
work, and it's a good excuse to involve Peruvian students.
Yeah, absolutely.
I think Grav, do you want to? Grav? Does he want to? Okay, I can read his question. So he said, nice talk, Pat and Amanda. My question would be similar to pollinators, visitation, and SC and SI indicate that genetic loci for nectar volatile production are still segregating despite the fixation of SC alleles, or is there some balancing selection that's maintaining those diversities of those metabolic reproduction resources?
I would say that's something we haven't looked into. Obviously, the pollen is the big reward for the pollinators in this case. So we've got the bumblebees doing the buzz pollination. The tomatoes don't really provide nectar, but there have been some studies, definitely not in habrochaites necessarily, looking at different volatile compounds. And it was something I was interested in exploring for a while, but I was talking to the metabolics or metabolomics facility over here about getting something set up, and it seemed not the easiest thing. So we haven't, I would say, not specifically looked at it, but I think just based, you know, I'm not a bee and I don't have the same sense of smell as a bee, but as a person, the wild tomatoes all smell very different, either than the ones from the northern part of the range that don't seem very genetically diverse. You kind of like just or brush against them, and they have very distinct and different smells. So in the leaves, at least. Florally, I don't think we haven't done a good job characterizing that. But do you have anything to add, Pat?
Not really, but I was very impressed. I mean, yeah, going into the field has really changed how I think about everything. But just the tremendous amount of pollinator activity in those self-compatible populations. I mean, the only place I really didn't see any activity was with Solanum neoricii, and it can barely make the effort to open its flowers. Even it's the only really autogamous species that I observed. But otherwise, there's lots of, I think, lots of outcrossing going on.
I agree. And it's something we haven't characterized well in our system. Like that's some field studies we proposed that didn't end up getting funded, but it was to look more at outcrossing versus selfing rates. Because the stigmas are exerted pretty far. And so, you know, just the likelihood that pollen's going to reach the stigma, self-pollen, and be able to go down and make self-fruits is, you know, questionable. We can like do some studies here in our fields at CSU, but it's not necessarily indicative of what's actually happening out there. And that's obviously like really important for this, you know, genetic structure stuff and how these things really are spreading. But I think there's a lot of evidence, you know, that suggests that, you know, maybe there's these like even years where pollinators have declines and things like maybe even in these mixed mating populations, if they can occasionally throw off self-fruit and like keep reproducing that way, you know, that could be an important mechanism for, you know, persisting in the face of a lack of pollinators, I guess.
Yeah. So we all need to take Mario with us on our next field visits.
Rimco just said great talks and that he can't find Solanum archicum anymore. I don't know if he has any more comments, but
at that same site, that is a shame. That is really, that's really too bad. Yeah.
Then Iris, so endosperm balance number could explain this
post-zygotic,
post-zygotic seed failure, like in potatoes.
Absolutely, absolutely. And let's see, I saw Thomas Stadler here. Yeah, so he could actually comment on that better than me. So I don't know if you want to answer that, Thomas.
Sorry, yeah, I can. Thanks. Yes, Pat knows that we happened to publish a review paper 2 months ago in Current Opinion of Plant Biology, summarizing both known molecular mechanisms, but also evolutionary drivers or hypothetical evolutionary drivers of seed failure, where the tomato system is one of the, of course, the drivers of empirical systems, but of course, it's all across the board. Of course, in Brassicaceae, Arabidopsis, and Capsella, there's more molecular work has been done in the past couple of years. But conceptually, this would be the uniting concept, endosperm balance number or effective ploidy. And in the beginning of your talk, Pat, when you described the phenotypes of the reciprocal crosses where the endothelium is much thicker in one direction of the cross, which we also have seen in Chilense peruvianum hybrids. I think if I remember correctly, if this is a paternal excess situation, so when the mother species, the maternal species is overpowered by the stronger father, this is the phenotype you get. Probably having to do with some mediating by auxin pathways that regulate the seed and endosperm growth.
Well, I'm especially fascinated by that because it's almost an evolutionary, it sort of looks like an evolutionary throwback to gymnosperms, which, you know, have this, the sporophyte is actually providing all of the nutritive tissue for the embryo. Anyway, I find that really, really interesting.
All right. I don't think there's any more questions or comments, but I want to thank both of you, Amanda and Pat, for giving your talk, and it was really interesting. And look forward to the botany section that both of you have put together.
Yeah, thanks for having us. Hopefully see you there.
All right,
virtually.
thanks everybody.
And
Bye-bye.
join in next week for Loretta Ferreira. She's going to be talking about magnificent wild petunias and their evolutionary history and genetic diversity. So thanks everyone. See you next week.
Thank you.